Imagine having, a highly advanced, incredibly powerful weapon against cancer. You deploy it, but there is a massive catch.
Right.
The tumor actually knows it's coming, and it actively builds a specialized shield to absorb the blow.
Yeah, which is a terrible scenario.
It is, but that is the frustrating reality of radiotherapy, right?
Yeah.
I mean, we rely on it constantly. It's a complete cornerstone of modern oncology, used in roughly half of all solid tumor treatments.
Absolutely. It's basically level 1 evidence standard of care for so many cancers.
Exactly. But tumors are, well, they're exceptionally adaptable. They often grow resistant to the radiation by recruiting this, army of immunosuppressive cells.
Yeah.
So they're thickening their defenses the more you try to blast them.
The clinical hurdle there is just immense because, we are essentially fighting the body's own biology.
Right. The body wants to heal.
Exactly.
Yeah.
Radiation causes DNA damage and cell death, which is the goal, of course, but the surrounding healthy tissue interprets that damage as a severe wound.
Like a massive trauma.
Yeah. Yes. So the environment floods with wound healing factors like, TGF-beta, which naturally attract regulatory T cells and suppressive macrophages to calm the inflammation down and literally repair the tissue.
So the tumor just hijacks this natural wound healing response.
Exactly. It uses it to protect itself from the very therapy that was meant to destroy it.
Which brings us to today. Welcome to our deep dive, everyone. Today, you and I are exploring a fascinating 2026 paper published in the journal Cancer Cell by Marco and colleagues.
It's a fantastic paper.
It really is. And it attempts to turn that exact wound healing response against the tumor itself. So our mission today is to explore this radical new framework that combines traditional radiotherapy with adeno-associated virus, or AAV, gene therapy.
And, they didn't just run a basic combination trial to see if 2 therapies are better than 1.
Right, that would be too simple. Okay, let's unpack this. The researchers uncovered that firing radiation at a tumor physically alters the tumor's internal DNA architecture. It essentially rewrites its epigenetic guest list to welcome a viral payload.
Which honestly runs counter to everything we assume about cellular stress.
How so?
Well, when a solid tumor cell is irradiated, it goes into a state of total panic. It halts replication. It shuts down non-essential processes to repair its DNA.
Right, it goes on lockdown.
Exactly. So it shouldn't suddenly become highly receptive to expressing a foreign viral vector.
But it does. And the delivery mechanism is the first major hurdle here, because AAVs are incredibly safe delivery vehicles, which is why we use them constantly in gene therapy, but they are notoriously bad at penetrating solid tumor cells.
They really are. They often just get stuck at the membrane, or if they do manage to get inside, the cell just silences them immediately.
So the analogy I like to use here to trigger some imagery is the Trojan horse. Think of the AAV as the Trojan horse, packed with soldiers, but it's completely locked out of the city gates.
I like that.
But the radiation acts like this massive battering ram that hits the gate so hard it accidentally alters the locks on the inside, letting the horse just roll right in.
That's a great way to visualize it. And the cellular mechanics making that happen are just elegant.
Break those mechanics down for us. What's actually happening to the virus inside the cell?
So when an AAV enters a cell, its genetic material doesn't integrate into our chromosomes. It forms a separate circular loop of DNA called an episome.
An episome, okay.
Right. And this episome just sits inside the nucleus alongside the host's chromosomes. Now, the researchers found that the massive DNA damage response triggered by the radiation inadvertently causes epigenetic remodeling of that specific viral episome.
Epigenetic meaning it changes how the genes are read, not the code itself, right?
Exactly. They observed 2 very distinct epigenetic changes. First, there was a significant decrease in DNA methylation on the viral genome.
And methylation is basically a biological stop sign?
Yes. Methylation acts as a restrictive brake on gene expression. It literally blocks the machinery from reading the DNA. So removing it lifts the silencing.
Got it. And the second change?
Second, they saw a sharp increase in a specific histone mark called H3K27AC.
Okay, let's give the listeners a quick mnemonic for that. H3K27AC. Think of it as highly active chromatin. The AC stands for acetylation, but active chromatin helps me remember what it does.
That's perfect, because histones are the proteins that DNA winds around, like thread on a spool. And that H3K27X mark acts as a massive README sign that actively promotes gene transcription. It unspools the thread.
So the virus is unspooled and ready to be read. And the data highlighting this shift is striking. They tested this in vitro using 2 very different cell lines.
Yeah, they used MC38, which is a mouse colorectal cancer, and RT112, a human bladder cancer.
Which is smart. Testing a mouse in a human line proves this isn't some quirky mechanism isolated to one species or tissue.
Exactly, and they loaded the AAV with a standard CAG promoter, basically just a generic, always-on switch, and a green fluorescent protein to act as a visual tracker.
Like a biological glow stick.
Right. When they irradiated the cells prior to introducing the virus, the infection rate just spiked. And the mean fluorescence intensity, like the actual brightness of the individual cells, was drastically higher.
Meaning the radiation was literally forcing the cells to mass produce the viral payload.
Precisely. And we can actually trace that mass production down to a specific molecular organizer. The radiation promotes the recruitment of a transcription factor called YY1 directly to the viral genome.
YY1. So what is YY1's job here?
It brings in the molecular machinery needed to physically transcribe the DNA, and the researchers verified this causality beautifully.
Right, they did 2 knockout tests.
Yeah. First, they mutated the specific sequence on the viral DNA where YY1 binds, which completely erased the radiation-induced boost.
Wow.
Then they treated the cells with A485, which is a chemical inhibitor that blocks the enzymes responsible for adding that activating H3K27 apsahistone mark.
And that also stopped the boost.
Completely neutralized it.
Wait, I do wonder, though, does irradiating a cell just turn it into a stressed-out sponge? Like, will it just mass-produce any genetic material you throw at it? What if we delivered, standard mRNA instead of an AAV?
That is a crucial question, and the researchers actually controlled for that exact scenario. They packaged mRNA into lipid nanoparticles, the same delivery mechanism used in the mRNA COVID vaccines, and introduced them to the irradiated cells.
And what happened?
Nothing. The radiation did absolutely nothing to enhance the expression of the lipid nanoparticles.
Oh, wow, so it's not a sponge at all.
No. The enhancement is uniquely specific to the biology of the AAV episome and how it interacts with the cell's epigenetic repair machinery.
Okay, so let's do a quick 5-minute recap for you listening to lock in these concepts. First bullet. Radiotherapy doesn't just kill cells, it alters their epigenetics. Second bullet. This alteration specifically unsilences AAV viral episomes by removing methylation and adding that active chromatin H3K27 exact mark. Third bullet. This process is driven by the YY1 transcription factor and is totally unique to AAVs.
A perfect summary.
So, question for you to ponder at home. If you can force a tumor to mass produce a viral payload just by shining a beam of radiation at it, what payload would you choose to destroy the tumor from the inside out?
Which brings us to their choice of payload.
Yes, because an empty delivery vehicle doesn't help us. The researchers chose a cytokine called IL-12. And here's where it gets really interesting, because IL-12 carries a massive amount of historical baggage in oncology, doesn't it?
Oh, massive baggage. IL-12 is often viewed as the holy grail of immune activation. I mean, it is a master regulator cytokine. It bridges the innate and adaptive immune systems.
It's basically the general of the immune army.
Exactly. It awakens dormant killer T cells, it activates natural killer cells, and it drives a highly aggressive anti-tumor response.
But the issue is, we've known about IL-12 for decades, but clinical trials using recombinant IL-12 have historically just failed miserably due to extreme toxicity.
Lethal toxicity in some cases.
Right, because if IL-12 enters the systemic bloodstream, it activates the immune system everywhere, all at once. You get fatal systemic inflammation.
So the researchers had to figure out how to confine this potent weapon strictly to the tumor.
Because if they used a standard promoter that just pumps out IL-12 continuously, and that virus happened to escape the tumor and get into the blood,
the results for the patient would be lethal.
So how do they engineer around that?
The engineering of the vector here addresses that spatial control perfectly through what we call a biological logic gate.
A logic gate like in computer programming.
Yeah.
If-then statement.
Exactly. Instead of an always-on promoter, they used an ISRE promoter, which stands for interferon-stimulated response element.
So if there is interferon, then turn on the IL-12.
You got it. The viral payload remains completely dormant until it is exposed to type I interferon,
which connects so beautifully back to the radiation. Let me walk through this mechanism because it's fascinating. When you blast a tumor with radiation, you shatter its DNA.
Right.
Those fragmented pieces of DNA leak out of the nucleus and float into the cytosol, the cellular fluid, where they absolutely shouldn't be.
DNA in the cytosol is a huge red flag for a cell.
Exactly. And the cell has an internal security system for misplaced DNA called the CGA sting pathway.
Yes, the CGA sting pathway is critical here.
So CGAS is an enzyme that detects that floating DNA. and it binds to STING, which sets off a cascade. When CGA STING detects those broken fragments, it trips the alarm, causing the cell to pump out massive amounts of type I interferon.
And the logic gate relies entirely on that sequence of events. The AAV enters the body and does absolutely nothing. It only activates its IL-12 payload if it finds itself in a microenvironment saturated with interferon.
And the only tissue saturated with interferon is the specific tumor that was targeted with radiation.
Exactly. It provides absolute, localized control.
But wait, there is a vital secondary safety catch to consider, right? Because the liver naturally produces baseline levels of interferon, and AAVs naturally pool in the liver because the liver filters the blood.
The researchers absolutely anticipated that liver leakage. They knew they needed a fail-safe.
So what was the fail-safe?
To prevent the virus from activating in the liver, they incorporated multiple binding sites for a microRNA called miR-122 into the vector.
miR-122.
Okay. Now, microRNAs are these tiny molecules that naturally block gene expression by degrading messenger RNA. And miR-122 is expressed almost exclusively in liver cells.
So it's the liver's master off switch.
Right. If this AAV accidentally ends up in the liver's own miR-122 binds to the viral RNA and acts as a strict permanent off switch, completely neutralizing the IL-12 payload, regardless of the interferon levels.
That is just brilliant. And the comparative data on this safety profile is incredible. They engineered one AAV with an always-on promoter for IL-12 and compared it to their smart logic-gated version.
The results were night and day.
Yeah, when injected into mice, the always-on version caused severe fatal weight loss, completely mirroring the toxicity seen in those early human trials.
But the mice receiving the logic-gated AAV stayed completely healthy. Their tumors were eradicated, and blood analysis showed IL-12 levels were sky-high inside the tumor tissue, but practically undetectable in the peripheral blood serum.
It is a profound demonstration of synthetic biology. Okay, let's do our second quick recap. First bullet, IL-12 is highly toxic and systemic, so it needs tight control. Second bullet, they use an ISRE promoter, a biological logic gate, that only turns on in the presence of interferon created by the CGA STING pathway after radiation. Third bullet, they added a miR-122 off switch specifically to protect the liver.
Spot on.
So, question for you listeners, how does the tumor microenvironment actually change once this localized IL-12 bomb goes off?
Because it's not a subtle change.
Right. We discussed earlier that radiation usually brings in a suppressive shield of regulatory T cells and macrophages to heal the tissue. How does this single localized burst of IL-12 override that deeply ingrained biological response?
We can see the override clearly in the single-cell RNA sequencing data.
Single-cell RNA sequencing, meaning they're looking at the exact gene expression of individual cells one by one.
Exactly. They analyzed thousands of individual immune cells within the tumor microenvironment, or TME. And the IL-12 doesn't just cause a minor shift in the immune cell populations, it orchestrates a complete hostile takeover.
Let's look at the CD8 plus T cells, the primary assassins of the immune system. The shift there is dramatic, isn't it?
Very. In a standard irradiated tumor, the few CD8 T cells present are usually exhausted. They are suppressed by the tumor's defensive shield.
Just tired and ineffective.
Right. But the single-cell data shows the IL-12 burst triggers a massive influx of new CD8 T cells, forcing them into a highly proliferative effector state.
They wake up and start dividing.
Yes. They immediately start producing granzyme B, which is the protein used to induce cell death, and they secrete their own interferon gamma to keep the environment inflamed and active.
But even more fascinating to me is what happens to the tumor's own shield. Solid tumors rely heavily on myeloid-derived suppressor cells, or MDSCs.
And a specific type of macrophage that expresses arginase 1, which suppresses T-cell function.
Right, the ARG1 plus macrophages. But the single-cell RNA data revealed that the IL-12 physically forces these suppressive cells to repolarize.
They essentially abandon their protective role.
They just switch sides in the middle of the fight.
They do. They transform into pro-inflammatory monocytes and macrophages. And once they flip, they start secreting chemokines like CXCL9 and CXCL10.
And what do those chemokines do?
They act as chemical homing beacons, signaling to the rest of the immune system to send even more killer T cells to the tumor site.
It's like calling in air support. And the physiological changes extend beyond the immune cells themselves, right?
Yeah, the endothelial cells lining the blood vessels inside the tumor drastically alter their properties too.
How so?
They upregulate the expression of a molecule called ICAM1.
ICAM1. I like to think of ICAM1 as molecular Velcro.
That's a perfect analogy. As the newly activated CD8 T cells rush through the bloodstream, this ICAM-1 Velcro along the vessel walls grabs the T cells and literally pulls them directly out of the blood and into the tumor tissue.
That's incredible. Okay, time for our third recap. First bullet. IL-12 floods the tumor with fresh, aggressive CD8 plus T cells producing granzyme B. Second bullet. Suppressive MBSCs and macrophages are forced to flip sides and become pro-inflammatory. Third bullet. Blood vessels put up ICAM-1 Velcro to pull more T cells into the fight.
It's a complete rewiring of the tumor from cold to hot.
So clearing a localized tumor with this coordinated attack is a massive achievement. But, and here's a big question for you listeners, what happens to the cancer that has already escaped? Because the deadliest aspect of oncology is metastasis, when the cancer spreads to the lungs, the liver, or the brain.
Right. Primary tumors rarely kill. Metastases do.
Exactly. So the researchers needed to know if treating a single accessible primary tumor could generate an immune response strong enough to hunt down distant metastases. So what does this all mean for metastatic disease?
They were looking for the abscopal effect.
The abscopal effect. Explain that for us.
It's a phenomenon where localized treatment triggers a systemic immune response that clears untreated tumors elsewhere in the body.
It happens occasionally with standard radiotherapy, but it is incredibly rare and, frankly, totally unpredictable.
Right. So to test if their logic-gated AAV could reliably induce it, they utilized dual flank models. They established tumors on both the left and right sides of the mice.
And they irradiated and injected the AAV into the right tumor only, leaving the left tumor completely untouched.
Exactly. They also ran a separate model where they induced secondary tumors in the lungs to mimic severe late-stage metastatic disease.
And the outcomes were just staggering. The untreated distant tumors in the dual flank model shrank dramatically, and the lung metastases were completely cleared.
The localized burst of IL-12 educated the immune system so thoroughly that it created a body-wide patrol.
Furthermore, they discovered that if they applied a very low non-lethal dose of radiation to those distant tumors, the systemic clearance was magnified even further.
But the mechanism driving the systemic clearance is where the paper really challenges fundamental assumptions in immunology.
This is the big twist.
It really is. Traditional models dictate that for T cells to hunt down a distant tumor, they must rely on antigen-dependent cytotoxicity.
Meaning, the T cell has to recognize a specific mutated protein, an antigen, that is displayed on the surface of the cancer cell via an MHC-I molecule.
Exactly. It needs to see the cancer's specific mugshot.
Which is exactly how cancers evade the immune system as they metastasize. They constantly mutate, downregulating their MHC-I display cases so they become literally invisible to circulating T cells.
Right, they take down the mugshots.
If the cancer cell hides its antigens, standard T cells simply float right past it. So I would naturally assume a therapy this effective relies heavily on preserving that antigen recognition.
That is the logical assumption. But the researchers proved this therapy works completely independently of standard antigen recognition.
Wait, really? How did they prove that?
They use 3 highly specific knockout models. First, they use mice completely lacking perforin.
And perforin is the primary protein CD8 T cells use to physically punch holes in target cells.
Right. And even without their primary weapon, the therapy still cleared the tumors.
Wow.
Second, they utilized BATF3 knockout mice.
Okay, that model is particularly crucial. Listeners, BATF3 knockout mice lack the specific cross-presenting dendritic cells required to chew up the tumor and present its antigens to the T cells in the lymph nodes. If you lack those dendritic cells, the T cells never get the mugshot of the cancer they're supposed to hunt.
Exactly. And yet, the therapy still eradicated the tumors in those mice.
Unbelievable.
And to push it to the absolute limit, they genetically engineered the tumor cells themselves to lack MHC-I entirely.
So the tumors were essentially invisible to standard T cell receptor recognition.
Completely invisible. And the systemic clearance still worked flawlessly.
But wait, if the T cells don't have their perforin hole punchers, and they don't even know what the tumor looks like, how on earth are they triggering cell death?
The clearance relies on a distinct secondary pathway driven by a mechanism called FAS-mediated cell death.
FAS-mediated. FAS.
Yes. The initial localized treatment creates a massive flood of systemic interferon gamma. That interferon gamma, combined with the low-dose radiation at the distant sites, physically forces the metastatic cancer cells to express FAS receptors on their surface.
And FAS is universally known in biology as the death receptor.
Yes. It is a biological self-destruct sequence built into the cell.
Okay, so the tumors are forced to display the death receptor. Then what?
Meanwhile, the systemic IL-12 activation causes the circulating T cells and natural killer cells to express extremely high levels of a complementary molecule called FAS-ligand.
Oh, I see where this is going.
Right. Because of this dynamic, the T cells don't need to recognize a specific mutated tumor antigen. The tumor cells are forced to display universal self-destruct buttons, and the activated immune cells are circulating with the specific molecular detonator.
The FAS ligand.
Exactly. They simply bump into the cancer cells, the FAS ligand binds to the FAS receptor, and the cancer cell cleanly initiates its own apoptosis.
It perfectly bypasses the most complex immune evasion tricks a metastatic tumor can deploy. That is just astonishing.
It really is a paradigm shift.
Time for our final mini recap. First bullet. Treating one tumor cleared distant lung metastases through the abscopal effect. Second bullet. This worked even in tumors that were invisible to standard T cells because they lacked MHC-I. Third bullet. It relies on FAS-mediated cell death, where the immune system just triggers a universal self-destruct button on the cancer cells.
Beautifully summarized.
By using the epigenetic side effects of radiation to unlock the AAV, they've turned a standard century-old treatment into a trigger for advanced gene therapy. And, the modularity of this platform is also a huge takeaway for me.
Oh, absolutely. While IL-12 was used here because of its raw potency, this radiation-activated logic-gated viral vector could be loaded with entirely different therapeutic payloads.
Like what?
Well, payloads tailored to specific cancer profiles, or it could be combined with existing checkpoint inhibitors to amplify the response even further. The possibilities are really broad.
It totally reframes our entire approach to the immunosuppressive shield tumors build. We aren't just trying to break through it anymore. We are literally using the shield's own internal alarms to smuggle in the payload.
We are turning their biology against them.
Exactly. So I want to leave you, our listeners, with a final thought to mull over today as you process all this incredible research. If a single, localized treatment just firing radiation at one spot can train the immune system to hunt down distant invisible metastases using universal death receptors, how might this completely shift the paradigm for incurable stage 4 cancers?
It's a profound question.
Right. Imagine a future where a surgeon doesn't need to painstakingly track down every microscopic metastasis in a patient's body. Instead, treating one accessible tumor could act as a customized full-body vaccine, waking your immune system up to clear the rest of the disease entirely on its own.
The integration of these distinct biological disciplines, radiotherapy, gene therapy, and immunology, certainly brings that future much closer to reality.
It really does. Thank you so much for bringing your expertise today. And to you listening, thanks for joining us on this deep dive. We'll catch you next time.